So welcome to video 5 in our course G07 on phylogenomics. Here we're looking at substitution models and how we can compare different substitution models. So let's think about the terminology associated with this area. So firstly, we have these substitution models, which are continuous time Markov chain probabilistic models describing the changes between the different nucleotides or indeed amino acids over time. So for amino acids, obviously that's more complicated. For nucleotides, it's more simple. But the principle for both is very similar. We can think about homogeneous process models associated with this, where we assume, for example, that the same substitution rates are operating across alignment sites, taxa, and time. We can also think about introducing compositional heterogeneity into this process and think about variation in nucleotide or amino acid frequencies across lineages of phylogeny, kind of extending the homogeneous process model initially. And an example of that is a Kimura two-parameter model, K2P, as you might hear it referred to. A complication with this is called heterotaxy, and that's where we get variation in substitution rates and composition at certain regions of the genome and our alignment of interest. So we have to kind of think about how complex we can make these substitution models. That also informs how processes are operating at rather over complicating things. One consideration for this assessment is that if you imagine that you're looking at DNA sequences or RNA sequences in a virus, you may get this issue of mutations varying quickly. And therefore, our observed genetic differences that we see in our sequences may not reflect the true ancestries. So you know that, for example, that ATCG substitutions divide into the two categories of CT that are transitions and AAG that correspond to transversions, of course, including uracil with the transitions as well. And therefore, we can correct this genetic differences for the portion of true differences that are likely to have occurred. So if we look at the juice and Cantor regression as the first time this was really developed, they can scale the proportion of observed differences P and obtain from that the true genetic distance inferred. So P is the portion of two sites differing, and D is the portion of two sites differing. P is the portion of the observed sites, so we're able to calculate D from this formula. And this tries to circumvent our issue of multiple substitutions that we may see A and C between two different sequences. But what has happened actually is that A has switched to T, G, and then T, and then A. This means that when we look at the proportion of sites that are different, the genetic distance estimate from is a decent proxy of that, but it diverges rapidly once the portion of sites that are different is very, very high. And just aligning closely related sequences is more easy. Aligning very divergent ones is not as easy at all. So there's different substitution models. Starting with that, the differences between transitions and conversions, which is incorporated into the Kimura two-parameter model. We can also think about GC content as well that vary across different species. Like Mycobacterium tuberculosis has very high GC, whereas Plasmodium falciparum has very low GC, is very AT rich. And that's why it's hard to sequence Plasmodium falciparum. And we also are aware that many genes have regions that are highly conserved or indeed that evolve quickly too. So where possible, what we can do to combat this is use smaller genetic distances they're more reliable if that's possible. Larger genetic distances are less reliable because we'll have this saturation effect on multiple substitutions. The divergent sequences are more affected by the substitution model choice, and therefore it's more important if you have more divergent samples being examined. Gaps are typically ignored. And it's possible then if you have an appropriate substitution model and the molecular clock signal is strong, then you can estimate divergence times. However, we have to be aware that these two key factors are important on that. So if we assume compositional homogeneity, if the samples are very divergent, they actually have different rates of change at different genomic locations and also different probabilities of changing from A, C, G, T to each other. And therefore, we're comparing apples with oranges. So we know that samples that have intrinsically similar base compositions will cluster more, independent from their genetic distances. So what better models do is they let the base frequency, the composition vary across the different tree. Challenge in that area is that this obviously computationally slow. Another option then is to do something like IQT does, which is to remove regions lacking composition heterogeneity. Obviously, that loses data, so it depends on what's important for your particular research question. We can also aggregate different character states. So we can compare, say, rates for purines and pyrimidines, and encode these as binary characters and do similar for amino acids that have similar properties. However, with that, we also have to consider codon usage bias, which will affect the rates there. And therefore, if that codon usage varies across the different samples in the same phylogeny, we have to consider how that might affect the rates of change observed in the tree. An example of how that might be important is, say, for glycine, where it's GGN. So the third position can be any nucleotide, GGA, GGC, GGT, GGU, GGG. And therefore, we can consider then, if we look at this example down here, where we have variation across this different tree, we have more mutations at the red end and fewer at the blue end. We can see that certain mutations shown by circles have been accumulated at the fast evolving sites, which creates more instances of what's called homoplasy. So this is independent creation of the same allele that appear identical, but actually have different evolutionary origins. That can be problematic in terms of inference, because it may bias our interpretation of the data. And then we have regions like this that are very, very pointy mutation. We see that there are certain changes that appear to be shared, but we have to kind of model as to how accurate that hypothesis might be. So we can think about this where you have human Drosophila, and then two other divergent organisms here, comb jellies, and what one is this? I wonder. We have a two tree. We have a tree inverted with a single rate and then a variable rate. So you can see that the mutations are placed on this tree depending on what the hypothesis is. So if you think about it, I'm a homogeneous rate model. We may get an underestimate at fast evolving regions. This may underestimate the convergence chance of different mutations. If we look at a heterogeneous model that allows rates to vary across different sites, we may be able to get a better tree than it infers where these mutations arose. So if we look at the alignment, we'll see more stars occurred on these two lineages that are at the occurred along these lineages. If we consider then another example on the next slide, where we have an alignment shown as the one below, we see that there are certain regions with more mutations that are parsimony informative, and then other regions where we see other mutations that have occurred. We consider the chances of mutating between different amino acids and the rates of change across the whole alignment. So if you look at the composition of the different amino acids in this alignment, we see that there's no differences. However, when we look at different regions, we can see that some have overrepresentations of certain amino acid types compared to other ones. So this means we have to think carefully about a model of what particular amino acid is actually possible to occur at a particular locus. So if we have a homogeneous model ignoring site composition heterogeneity, such as this one, we underestimate the change expected for regions with restricted compositions, and this underestimates the chances of convergent changes. Heterogeneous site models allowing this site composition variation is more likely to get the true tree. So what we can do in terms of models that we use is employ what's called a gamma distribution. So it's an add-on to an existing model, sometimes written as plus here plus gamma or plus G for gamma to the substitution model. So we can have a G-Cantor 69 model with a gamma distribution of sites, site variation rates. And same for the general time reversal model as well. Another option is a free rates model, assumes a few discrete rate classes. And then we also have gamma mixture models, assume we have more than one gamma distribution as well. You may also come across cat models, and this is where we have the size weights are distributed according to a mixture of k to different categories. So the number k would vary depending on what the actual alignment. And that's where we have these different categories come from the substitution matrix Q, and the class to which each sites belong is from the allocation of some variable Zi, which comes from these categories we have there. So an example of this is one which we've come across before. It comes from the first and second half of the alignment having very, very different patterns, where we have A closer to B, but then we have C with D, and this is what's called heterotypy. So we have this first half are B and D are more similar. The second half, we have A and C are more similar, and therefore we end up with like this. We have long branches for A and C for the first half, and then for B and D in the second half. And therefore, an appropriate inferential model to handle this needs to consider longer divergences between, say, A and B versus, A and B versus C and D. So you can see that we've got a faster rate and a slower rate as well. So what's shown is the simulations for 5,000 sites for each partition. There's two different partitions here, assuming it includes Cantor model based on IQ tree. And this tree is essentially the average of these two regions. What we see there, we have A and C erroneously joined, and B and D are also erroneously joined when we know that, in fact, for each individual segment, A and B are closer to each other relative to C at least. So we also have transitions versus transversions, and that's incorporated into things like the general time reversal model. And that's assuming each of these four nucleotides has a different frequencies estimated as the main rate plus 3 free rates for each of the four nucleotides. We also add on to this 6 different substitution rates for the changes from A to G, C, T, and then G to C, G to T, and T to C. Obviously, for amino acids, there's more parameters and that gets more complicated. The central idea is that obviously transitions and transversions are chemically very different. You've got A here with 2 big rings, G with 2 big rings, so it's much more easy to transition between either of these. Same for C and T. That's 2 mutation options there for those two. And so as conversions, we've got to change from A to C, G to T, and also G to C and A to T. So that's 4. So you'd expect relatively for unconserved sequences where it doesn't matter what nucleotides presence have a ratio of transitions to transversions of 2 divided by 4, so 0.5. What we're observing in practice is that the rate of transitions versus transversions will be something like 2 would be more observed or higher. You could get sometimes 3, and that's because C and T chemically much more similar to each other, and vice versa for A and G. So they're much more tolerated in terms of the structural integrity of the nucleotide sequence that's present. For amino acids, there's a whole field on this that we don't go into here, but empirical estimates are used, but that depends on the organism that you have of interest. It depends on the sequence type as well. Viral, chloroplast, mitochondrial, and so on are all very, very different. So then for our genome-wide SNPs, we think about using partition models to accommodate this heterogeneity across the genome. Certain regions do this, like IQ3 model test in G, which we'll use later. And partition models are generally better, as you can understand. So we had these homogeneous partitioned and mixture models. They assume that any site for a lambda size X evolves independently and estimates the probabilities of having a particular allele at a particular site as a product across all the different sites. So mixture models calculate the chances a slice belong to a partition. They may use multiple substitution matrix. For example, with the cat model, it treats the mixture components as a free parameters and estimates the mean of those frequencies and mixing portions from the alignment input. So if you look at mixture models further, they assume the sites in the alignment are a mixture of m classes. We don't know in advance which class a site comes from. So it might be 1 over m at the start, and therefore it's trying to assess across these 8 sites which model is most relevant here. So it's looking at the probability of observing the data at site I as an average over the m site classes. So we're summing across for each of the sites, 1 to, so the classes 1 to m, the weight divided by the probability of observing that allele given the model at hand. For site homogeneous models, these are simpler and they assume the same substitution rate and process across all the sites in the alignment. So that's looking at the probabilities pi of each site xi, and that's under a shared model M1. So those kind of site substitution models allow us to think about different substitution models and how they work. So the one I mentioned before was the Jukes-Cantor model, where it assumes that all the nucleotide changes have equal chances, but we know that a gamma distribution can be added. That's taking P and getting D from that, the true number of differences. K2P parameter allows transitions versus transversions to vary separately. And the Jin and Nye extension of it allowed for variation across sites through a gamma distribution. So this more advanced formula down here. Then we have the K81 model, which is an extension of the K2P model. It allows different rates for 2 different transversion classes, for A to C versus C to G versus the other ones. This is known as the 3 substitution types model. We also have the Felsenstein 1981 generalized Jukes-Cantor model, which relaxes the assumption of equal base frequencies. Related to this are the K80 model, which relaxes an assumption of equal base frequencies as well. And then the T92 model is the Tamura 1992 model, generalizes the above K80 model by relaxing the assumption of equal base frequencies by taking into account GC bias. We also have the TN93 model, where that Tamura Nye extended that existing model, where they have different rates for 2 kinds of transitions, A, G, and C, and T versus the other transversions, and the base frequencies are assumed to vary as well. We can add a gamma parameter to this as well. You might use, you would probably use the generalized time reversal model, which allows all the rates and frequencies to vary a lot. This is the most general model, and it has 6 substitution rate parameters and 4 equilibrium base parameters. Which these are the main one, and then the 3 other options. So we can summarize that for DNA based on this distribution, where we have rates for A to A, C to C, G to G, and T to T down here. And what we're thinking about is the substitution mu, and then we have the rate parameter for each possible transformation of one base to another. And then we have pi is the frequency of the different bases of C, G, T, or A as appropriate. And then we can think about the probability of changing, say, from A to a G, G to an A. So we can look at the chance of certain nucleotide changes over time, and we can estimate that by scaling the Q matrix by this formula, where if the branch length B, that's formed as an exponential, and that includes things like modeling that an A to A change, which is the same base, is actually a transition over time. So shorter time frames increase the probability that it'll stay in the same state, whereas longer time frames mean transitions are based on the frequencies observed. There's a cartoon of these models, where we have our Q matrix here at the top, And then we have the different rates here for A, C, G, and T that sum to 1, which allows us to infer 3 free parameters in the main one. Then we have the different probabilities of changes here for 6 different models, which can be inferred from the data as well. So you can see that these are different substitution models and the assumptions that they make. A tool that we will use later is model test. So model test allows us to compare different substitution models based on these criteria. So we look at hierarchical likelihood ratio tests, dynamical likelihood ratio tests, the AKA information criteria, Bayesian information criteria, and decision theory as well. This also determines the uncertainty parameters for anthropologies. So if we look at the substitution models that are possible, we have them listed here. So you got the Juice Cantor model, the Felsenstein 1981, K80 Kimura model, HKY are among the most popular ones. There are more models for amino acids, but they are not shown here. And for each model, it's looking at a fixed maximum parsimony topology, but we can use maximum likelihood in this as well, which might be typically more preferable. So its criteria are based on these different factors, that the likelihood ratio test is based on minimizing type 2 errors. The ARC approach is for minimizing the Kullback-Leibler distance. The ARC C model is based on the same thing. BLC is a Bayesian approach based on maximize the posterior identity. And then we also have decision theory as well. So if you think about the Kullback-Leibler divergence, that's basically essentially looking at the differences between two different distributions. That is the sum of the area that is present in the blue distribution that's absent in the red distribution shown over here. So the AIC reflects that relative difference based on observed parameters. And it's calculated by the AIC is -2 by the log likelihood of the model plus 2k, where k is the number of parameters. And what we want to do is minimize the AIC. So that means that the parameters will have a small effect on this and should improve the model. If not, they'll actually, if they don't improve the model, there'll be a penalty as well. So what we can do is get the corrected AIC, called the AICC, to adjust for the sample size n. So you can see n is incorporated there. So if we have a higher n, that will offset the effect of the parameters. As the sample size increases, the AIC will approximate the AICC will approximate the AIC model. Other options are the Bayesian information criteria, which is penalizes the sample size n, depending on how many we have. And then we also have decision theories, which ranks the models by the weighted expected error in the branch length estimates. So that's obtained from the BIC model. So what we can do for the model test NG is perform a likelihood ratio test, LRT. So for example, if we've got two models here, we can consider the data D in the model MI, where we might have two models, I is equal to 1 or 2. And therefore, what we can do is think about the Bayesian probabilities. It simplifies as simply taking the likelihood of model 2, the alternative model, versus the null model 1. So that's our starting point model at the start. And we're comparing them as a ratio, taking the log of that ratio. By taking the log of a ratio, we simply can subtract one from the other, which makes it more easy to calculate the likelihood ratio test. We then take that value and we compute the difference in the degrees of freedom that's present between model 2 and model 1. Model 2 will presumably have more parameters than model 1, a more simple model. If we take an example here, where we're computing this formula, so we're taking twice the log likelihood of the difference between them and compare that to a chi-square distribution. If we get a result like this, where we get the HK85, HKY model 85 has this particular likelihood, we can compare that to the GTR model and see that it's that the likelihood is different between them. The HKY 85 is higher, but we have to consider whether it's actually better. So we can use this formula by putting in the likelihood for model 2 in there, likelihood for model 1 in there, taking twice the difference and comparing that to quiet distribution to see how extreme that is. So what we see is that this is not sufficiently extreme required for to have a p-value less than 0.05. And we use this function in our p chi-squared, taking in an argument, a number of degrees of freedom, and a criteria to estimate the p-value effectively. So that's how we can link the model test NG to our usage of R. So please see the next video where we'll extend this when we think about tree routing.